I would like to thank the organizers for the invitation to speak. What I would like to do, once I get this open, here we go, is try to vertically integrate the talks that you've heard up until now. I think in some cases when we did get started 10 years ago, talking about gene-environmental interactions in autism risk was viewed, one, as heresy, and two, as hand-waving, because we had no real roadmap of examples where the gene-environment interactions might occur. So I'd like to give you two examples and then give you a third example, which to me is probably the scariest. So it's not surprising if you step back away from autism that, in fact, we know there are environmental impacts on outcomes for children's development. This is a National Research Council report about 8 years ago, which said basically concluded 3% of all developmental defects are attributed to exposure to toxic chemicals, and that 25% are probably the result of an interaction between genetic susceptibility, that is, inability to repair harm, or in fact higher sensitivity, and environmental insults. Now, this is primarily with an outcome of gross neonatal defects. So you can imagine that subtler effects on behavioral outcomes could in fact be higher in terms of gene-environment interactions. We think of autism and we think of mercury and we think of vaccines, but in fact we don't think that environmental exposures are putting us at risk simply because there's somebody out there marshaling what gets into the homes or what's applied on crops or what side stream exposures there are. And in fact, these are industrial chemicals, pesticides, ingredients, food additives. You've heard many of them. The vast majority, and I think most toxicologists will concur with this conclusion, the vast majority of commercially important chemicals lack adequate toxicity testing. And in fact, in particular, they lack adequate developmental neurotoxicity because we just don't know how to deal with it. Can you imagine putting 86,000 chemicals through a 2-year rodent study or a 2-year dog study? This is just inconceivable at the present time. We cannot do this. So let's think about how autism may confer susceptibility. To a toxicologist, the dose makes the poison. In fact, we know that atypically developing populations, such as genetically diverse populations such as human, have resistant individuals and hypersensitive individuals. And this is primarily due to their previous exposures and also their genetic makeup. We see this kind of variation even in the most highly inbred animals, such as lab rats and lab mice. Let's assume now that you've incurred 5 or 6 genetic polymorphisms or mutations, which in fact begin to shift your sensitivity. I'm going the wrong way, sorry. So if we think of autism as a diverse, ideologically diverse neurodevelopmental disorder, we might think that there are actually individuals that respond in an adverse way at much lower concentrations of a hypothetical toxicants. But if we could subdivide these or stratify these according to their particular etiology, their particular cadre of genetic susceptibilities, what we might find is, in fact, that there may be a subpopulation, it may be a small subpopulation, maybe only 2 to 5% of susceptible individuals, autistic individuals, that respond decades or even orders of magnitude at lower concentration in an adverse way. So typically the way our regulatory agency deals with this is they essentially set a no effect limit and then divide by 10 for neonates, and then divide by 10 the dose again for susceptible populations, which gives you about a 100-fold margin on average. This may not be enough for children with autism and other developmental disorders. So as you've heard, we know that autism has high heritability, but may also have high sensitivity to environmental modifiers. Multiple genes confer risk. We heard estimates from anywhere between 5 and 100 different genes may in fact be contributing to susceptibility. We also know that there are DNA methylation and acetylation abnormalities in some autistic children which confer epigenetic mechanisms to susceptibility. And we now are pondering the possibility that de novo copy number variations, the very ones that make you and I different, can actually be even more different in some children with autism that, again, confer heightened sensitivity. So I'd like to give you some perspective. So could autism represent a multi-system disorder whose outcome is likely to be more profoundly impacted by environment? And Irva has alluded to this, that just because something has a 60 to 90% heritability does not mean necessarily that the environmental susceptibility is a small part of that outcome. So let's look at specific examples. One of the overarching hypotheses about what causes autism is an imbalance in excitatory and inhibitory circuits in the developing nervous system. Many genes that are shown here, such as PTEN, which you've heard before, MECP2, the Fragile X mental retardation protein, FMR1, neuroligin, these are all genes that actually regulate aspects of excitability in the central nervous system. When these are dysregulated, what might happen is that you change the connectivity of neuronal circuits, either making them more prolific when they shouldn't be or less connected when they should be. You can have an imbalance between excitatory synapses and inhibitory synapses, which would promote seizures and hyperexcitability. You might also have modulators such as serotonin, which you've heard about earlier, which tend to drive the nervous system to integrate in very awkward ways, which could produce things like obsessive-compulsive disorder. So it turns out that most heavily used pesticides that we've allowed to get to market, in fact, influence and are by a priori targeted to upset the very balance of the central nervous system. One example is cholinergic neurotransmission, especially the nicotinic type cholinergic transmission. Organic phosphate insecticides, such as chlorpyrifos, have been shown to decrease birth weight. In fact, chlorpyrifos was banned by the EPA for home use because there was decreased birth weight associated with levels of exposure. And we've heard about premature NICU associations with autism. Do we know that organophosphates promote or increase susceptibility to autism risk? I'd also like to point out that in addition to the primary mode of action that's been ascribed to organophosphates, that is acetylcholinesterase inhibition, there is now evidence, and you might hear of this a little later, that lower exposures, some much lower that inhibit cholinesterase activity, in fact influences signaling pathways which have been related to autism. These include an increase in the phospho-Krebs signaling pathways which have been associated with autism, and changes in fibroblast growth factor expression. Now, acetylcholinesterase inhibition is one way to get at changing cholinergic signaling. And organophosphates have been severely restricted in the homes. However, we now have a whole new set of insecticides which are in the home and are used broadly in the home. These are neonicotinoid insecticides. Instead of inhibiting acetylcholinesterase, they actually target the acetylcholine receptor directly. And when these were developed, they were developed because they tended to act on the insect receptor and not so much on the mammalian receptor. The problem is that in the days when these were being developed, we knew only a couple of gene products that contributes to nicotinic cholinergic receptors in the mammalian brain. We now know of all of these. And some of these have in fact been implicated as being defective or deficient in their expression in autism, and I've given you the references here. So one way in which kids could be hypersensitive to neonicotinoids is simply because they don't make as much or sufficient levels of nicotinic cholinergic receptors, especially if neonicotinoids target certain subclasses of these receptors that we actually have no information on. Now, organophosphates insecticides, as you've heard from Craig, actually have been implicated in PDD-NOS. This is a small study, a Chamaco study out of Berkeley. This is a highly exposed group of individuals. They're pesticide farm workers. They're farm workers highly exposed to organophosphate pesticides. And this study was the first to actually show an increased risk through exposure of organophosphates and outcomes such as PDD-NOS, which may in fact be related to autism. There's another way that you can confer susceptibility to organophosphates. There is several papers out now that shows that peroxidase gene variants are associated with autism in North America. What is peroxidase? Peroxidase is in fact an enzyme which metabolizes organophosphate to what we think are inactive products. And in fact, the associations are made in the United States, but not in Italy, which suggests that in fact maybe, and this is one of the conclusions or speculations that were made in this study by the authors, was that the reason that the association is detectable in the United States is that during this period of time when they collected the data, organophosphates were actually used in the home, whereas in Italy they were not, they were banned. So here is a model where one can serve to do directed epidemiological or molecular approaches based on mechanism. We know that there are genetic susceptibilities in autism. This may arise from imbalances in cholinergic receptor expression. We know that there are organophosphates insecticides, which in fact inhibit those systems, well, at least the metabolism of the active neurotransmitter, and can enhance cholinergic transmission. If we think about PON1 polymorphisms that decrease the activity of the enzyme that degrades organophosphates, we can have a multivariate gene-environment interaction that could, in fact, lead to increased susceptibility. A second area that I want to point to is Gaba receptors, which have been implicated in autism. And Gaba receptor neurotransmission, especially of the GABA-A subtype, in fact, the targets of the chlorinated hydrocarbon insecticide. These include things like lindane, heptachlor, and endosulfan. They block GABA-A receptor transmission and alter their chloride movements of these receptors. So postnatal exposure causes hyperexcitability, and gestational exposure may cause depression. Well, these organochlorines, as you might have guessed, have been banned for many years now. There's still exposure to these compounds because they are extremely stable in the environment. So if we were to measure all of our bloods, we would have varying levels of these compounds. Here's one that's really potentially important. So we know that GABA-mediated neurotransmission is extremely important in a number of neurodevelopmental disorders. We have now a compound in the homes and now expanding in agricultural use that has limited toxicity testing. These are the 4-alkyl 1-phenylpyrazole insecticides, which include the lead compound known as Fipronil. I'm sure that most of you have come into contact with Fipronil. It is used widely in the homes for several products. Again, when Fipronil was developed, it was assumed to be insect selective. But when it was developed, we only knew a few of the gene products that code for GABA receptors in the mammal. We now know of 15. Could any one of these that have not been tested, in fact, be as potent a target for Fipronil as the insect receptor? And in fact, here's the scope of the problem. We have these chlorinated hydrocarbons. Lindane and endosulfan are still used. They block GABA receptors. Heptachlorochloridane and so forth are no longer used. They've been banned, and these are the years they were banned, but they're extremely stable, so there's still exposure. Fipronil actually acts at the exact same receptor, and the effects of these, in fact, are additive. It turns out that there's now evidence that one of the GABA receptors that's most prominent in autism, the GABA beta 3 receptor subunit, is as good a target for fipronil, endosulfan, and some of these other chlorinated organics as it is for the insect receptor. In other words, if you have 1 nanomolar potency at the insect receptor, we now know that you can demonstrate you have 1 nanomolar potency at the beta 3 receptor subunits. So there is no selectivity. Fast forward to about 2 years ago, could fipronil cause neurodevelopmental outcomes? Well, again, mouse studies have not actually been published in this regard, but there is a zebrafish study where a simple 24-hour exposure during critical period of development in the zebrafish causes tremendous amount of disorganization in the notochord and a very, very disruptive pattern of neuronal organization. Some would argue that this is a very high concentration, but I like to point out that this is actually a very short exposure. There are defective GABA A receptor genes identified in a subset of children with autism. These include epigenetic mechanisms, such as the MECP2 deficiency that has been associated with particularly downregulation of 10 to 15-fold of the beta 3 subunit of the GABA receptors. They're complex gene-gene interactions, and there also appear to be polymorphisms associated in the GABA receptor gene complexes in autism. So could children with heritable deficiencies in GABA A receptor-mediated neurotransmission be particularly susceptible to chemicals that interfere with GABA neurotransmission? And so I've given you some insight about chlorinated hydrocarbons, fipronil-like compounds which are increasing in use and sort of taking over for organophosphates, and some others which you probably have or have not heard about, such as the avermectins. Is there any epidemiological data that would suggest that exposure to GABA receptor blockers in fact increase risk for autism? And as Craig showed with the study in California matching database, pesticide exposure database with risk, in fact, if you look at the upper quartile, there's about a 6-fold increase in risk of having a child with autism, and this was a dose effect. So in other words, as the women were farther away from the exposure sites, the risk declined, which suggests that there may be a cause and effect. It's a small study, it needs to be replicated, but points to a sort of hypothesis-driven, testable theory, testable hypotheses on how you might proceed to look for gene-environment interactions. I want to finish by, you've heard about brominated flame retardants and their likelihood to be similar to polychlorinated biphenyls, which were banned in the mid-70s. Polychlorinated biphenyls are organochlorines. We think of them as being dioxin-like in some cases. It turns out that most of the chlorinated, polychlorinated biphenyls that we're exposed to today and that are in human tissue do not act like dioxin. They act like non-dioxin, and these have in fact been associated with neurodevelopmental disorders. If I was to give you a sort of measure of what fraction of non-dioxin-like PCBs are in a typical sediment, let's say in San Francisco Bay or in tissues from exposed populations, this would be the type of chemical that would be in the blood. This is important because these are the ones that are regulated. We do not regulate based on these chemicals. Why is this important? We've banned these. The general concentrations are going down in the environment. Well, it turns out that we actually haven't used our own precautionary principles. Brominated diphenyl ethers are non-coplanar compounds. So the neurotoxic effects of these non-coplanar PCBs probably is very similar to the mechanism of the PBDEs that Irva was telling you about. So PBDEs are intensively being studied now, but what about other chemicals in the homes that just fell under the radar screen? One such chemical is triclosan. It's in toothpaste, It's in antibacterial soap. It's in underarm deodorant. It's in probably 100 different compounds. When we started to look at this at Davis, a number of us said we would not find activity because these are directed toward bacterial cell walls, so that the target shouldn't be found in mammalian systems. Again, triclosan was screened for dioxin-like activity, and it has very little, very weak dioxin-like activity. It turns out that the precautionary principle should have been applied at the level of how stable the compound is and whether it bioaccumulates. So very recently, there was an article in Environmental Health Perspective, in fact last December, from the CDC scientists, which actually looked at levels of triclosan in human blood and urine samples. And of 2,500 people that were assessed, ranging in age from 6 years and older, what they found was, in fact, there was measurable and relatively high levels of triclosan in the blood. And these are some of the data. So there's huge variation. The variation is primarily explained by SES. The cleaner you are, the higher your blood levels, and the higher the exposure. And you can see even 6-year-olds to 11-year-olds have 5 to 6 parts per billion in the blood, and some of the higher levels were up around 40 to 50 parts per billion, which is relatively high. Why should we be worried about triclosan? After all, it targets bacterial membranes and not mammalian cells. Well, it turns out that's not true. Here are some excitable cells which are exposed for a very brief period to triclosan. And what we've done here is we've asked the cells to do something. So we're stimulating these cells at about 1 hertz, and the cells respond almost like clockwork. These cells are designed to do this. We begin to perfuse triclosan onto the cells, and what you start to notice very soon after the application is you get ectopic responses. In other words, responses when there shouldn't be responses. Again, suggesting a dysregulation of signaling. This happens to be a measurement of calcium signaling in these cells. 100 nanomolar causes ectopic responses, whereas if you go up to 750 nanomolar, you begin to deviate from recovery of the signal that's shown by this increased baseline, and in fact, in time, the signal fails completely. And so these are rather potent effects, which we don't know what they mean in terms of neurodevelopment. A graduate student in my lab has started to look at how this affects dendritic growth and complexity in culture, and there's rather some profound effects. Why should we care about calcium signaling in autism? Well, I've just given you a short list here in this table of genes that either directly dysregulate calcium signaling or are themselves directly regulated by calcium signals inside the cell. All of these have been associated with autism. These include MET, which you've heard about, the Timothy syndrome mutation, which is in fact a calcium channel, neuroligin, which you've heard about, is a calcium-regulated protein. So one possible convergence point is that, in fact, chemicals which disrupt calcium signaling, and there are many out there, can, in fact, be suggested to influence children with these kinds of polymorphisms and mutations more than they would the general population. So just as a parting framework, there are several ways in which one can study gene-environment interactions. We've heard epidemiological approaches, but I think we can also use mechanistic information gleaned from the genetics of autism, gleaned from what we know about mechanisms of toxic interactions from toxicologists, to come up with a model where genetic susceptibilities can confer much, much higher susceptibility than we ever thought to environmental exposures. And when the timing of the exposures is at a critical time in development, when those systems are being asked to do something, we can have an increase in autism risk, we can have an increase in severity, and we can also influence treatment outcomes. And so we think that autism susceptibility genes, in fact the ones we know of at the present time, can inform us about environmental modifiers of risk, severity, and treatment. Thank you very much.